Hello and good evening, and welcome to our session, Diagnostic Pitfalls in COVID-19. My name is Jacob Moran-Gilad. I'm the ECCVID Program Director, and I'm based in Israel. And my co-chair is Ed Kuipers from the University Medical Center of Leiden in the Netherlands. We have three fantastic speakers on three different aspects of COVID-19 diagnostics. And I thank all of them in advance for agreeing to share their experience stemming from very hard work done over the last months. And just with respect to our housekeeping, if you're not in full screen mode, you can see where you can upload your questions and submit them for the chairs to be able to pick them up. And I'll move on to the first speaker, Dr. Jim Hoggett. Jim is based in the UK, in LGC, which is the UK Institute for Measurement, and also in the University of Surrey. Jim is a microbiologist, and he's very much involved in metrology, the science of measurement. And his interest in life is measuring nucleic acids. And he does so also for COVID-19, but also to many other pathogens. So Jim will share with us his views on the analytical performance of PCR assays for COVID-19. Please.
Thank you very much, Jacob. Good day, everybody. I'm just going to try and share my screen. Are we okay? Excellent. Okay, well, thank you very much for the invitation. It's a great pleasure to be with you all. I'm now going to, for this talk, just talk about the analytical performance and some of the potential limitations of the SARS-CoV-2 molecular detection, and particularly the Rtqpcr, which is, bear with me, slides. Appears to, there we go. Which essentially is responsible for much of the diagnosis that has been used in response to finding the virus based on detecting the viral genome. The reverse transcription real-time PCR essentially performs over a period of cycles, and the fluorescence is measured in real time. And you can get different amplification curves. You can run calibration curves for this. And basically, the assumption along this, from the point of view of the quantitative bit, is that the blue amplification curve comes up earlier than the red one, and therefore there must be more nucleic acid at the beginning of the PCR. At least that's the underlying assumption. The units of measure on the right, they vary. Quantification cycle, cycle threshold, and crossing point are three that you will be familiar with. The cycle threshold, probably the most common. The quantification cycle, though, was introduced by the MICE guidelines to try and standardize the terminology to describe what it actually is. And this is the term that's used by ISO now in context of the real-time PCR. Crucially, however, the real-time PCR has the potential of being incredibly sensitive, even when measuring RNA as a complementary DNA, but it can only measure, excuse me, what you can get into the reaction. And things that influence that vary, especially in the case of SARS-CoV-2 molecular testing. The specimen needs to be sampled. Once you've chosen your specimen, the viral genome needs to be processed for purification, or at least the nucleic acid needs to be made available for the enzymes, and then you detect the virus. The discussions around how this whole process may cause issues are complicated by the fact that they're discussed at different levels. If you look at the literature, people will articulate different metrics considering the analysis only, which is the RNA and PCR step. But then there's the full analytical process, which includes the extraction, the lysis, to get the nucleic acid ready for detection, and then the full diagnostic process as well. Really, 1 and 2 are the only ones that are really interrogated extensively at the moment, largely because 3 is incredibly challenging to do so, to understand what's going on. And we haven't even mentioned the fact that the specimens change. So you can see all of these differences can lead to differences in the measurement. And the problem is, again, that the reports don't necessarily talk along the same level. I'm going to touch a bit on the quantity of the virus, and there's two arguments here. And I've been involved in some of the conversation, or listening to some of the conversations. And certainly in the UK, we've had the argument that detection is a simple process. The viral load is very high, 1 million copies per mil. The methods are highly sensitive. The genome presence, not the quantity, is what's measured. All of these are, in cases, correct. But there's a new narrative, particularly I hear on the UK, and that is that viral quantity measurements are needed. We need a quantitative cutoff to stratify patients to understand the difference between infected and people who are just carrying RNA and may not be infectious, sorry, they may not spread the disease, it may be gone and you're just measuring a signal from the past. And then the limit of detection is by its very nature a quantitative measure, which is an important metric for some of the processes like CE marking or even emergency use authorization in the US. If you look at this manuscript, it shows this quite nicely. As early, I think, this month. Basically, this figure on the right, I'm going to zoom in on, and it talks about the limit of detection and the differences with a number of the different methods that have got emergency use authorization. And the details for the purpose of this presentation are not necessarily needed. Suffice to say that there is over 4 orders of magnitude in the reported limit of detection of those methods. Now, I'm not advocating any particular limit of detection as being the required one. There's all sorts of discussions in the clinic about how this may be useful or not, having 2 sensitive methods. But the trouble is when we think about this discussion on the left-hand side about the RNA detection is a simple process and the fact that the limit of detection is a quantitative measure, that difference, that variation is going to influence the narrative of the discussion on how well a method that's measuring is impacting on the presence or absence of the virus when you're measuring it in a sample. I'd like to touch a little bit now on this need for a quantitative cutoff to stratify patients. Literature and reports talking about the fact that many of the methods may be picking up dead virus. And obviously, this impacts on the narrative along the lines of whether you're measuring a symptomatic individual, what are you going to do with them, versus screening and how you might manage that. There's a problem, though, and that's the discussion around the Qpcr. This is the method that appears to be discussed on how you might do those cutoffs, and particularly the cycle threshold or the quantification cycle, and whether we can have a particular quantification cycle of, say, 30. And if you look at the plot there, 30 would be on the right-hand side down here. And so what they're saying is that perhaps anything later than 30 should be considered as a negative. The trouble with this is that many people who work with Qpcr will know that it's highly variable. It's a precise measurement full of bias. And the data here, I'm just going to show you a brief outline. We're working on this data in much more detail, but I haven't got time today to go into this in detail. But if we were to apply quantitative cutoffs just based on the cycle threshold or the quantification cycle, and we assume that the data sets have 100% of the positives they were called, where you have no cutoff, if you have less than 30 cutoff, if anything that comes up after cycle 30 is not going to be considered positive, you now have 36% in the Great Ormond Street dataset are positive, 61% in the Belgian dataset, and 50% in the 2 assays used in Korea. So just that cycle 30 cutoff is going to give you different numbers of patients that are considered positive. Cycle 25 has also been described, particularly with some of the isothermal methods that are much less sensitive. Now, if the cycle 25 that the Qpcr is doing is being used to steer the limit of detection of other methods like isothermal strategies, then depending on which of these you use, it's going to influence how that works. And so you can see we're already getting disagreements just because we're using this Qpcr cycle threshold, or quantification cycle, as a cutoff. This is not really new, and it's shown nicely in some of the standardization work, equality control, external quality control assessments performed by the likes of Instand, where they compare the different data sets. And this is the April scheme that was used, one of the earliest schemes that was actually conducted to evaluate the performance of SARS-CoV-2 molecular testing. And if you just have a quick look at that, this is the range and the median quantification cycle. And if you just calculate those using the basic differences between the ranges, that varies between 100 and 10 million fold. And that's measuring the same tube. That's not considering any sampling, storage, transport. That is that stage 2 that I talked about, the sample processing, the RNA reverse transcription, and the actual molecular detection. And so we're getting between 100 and 10 million fold. Well, perhaps 10 million fold is an extreme, and I think there may be preamplification in there, but certainly 100, 1,000, this is normal. And the fact is it's not new. We've known this for over 12 years. Work by Jacqueline Fryer and Phil Miner at the NIBSC has shown this very clearly. When looking at non-calibrated viral measurements, you get vast differences in the quantification cycle. So nothing here is new. And we should be expecting this. We should be understanding this as a community, that just applying a cycle threshold, a quantification cycle cutoff is going to be variable. You might be able to do it in your particular laboratory with a particular number, but even then it will vary. Different people doing assessments of the where you would measure the cycle, it will vary. Even when you're doing the same experiment, you get a new batch of probes in, it's going to move that quantification cycle measure. And so it's a very precise but biased measure. And so we need some kind of calibrator if this is ever going to work. And consequently, the statement that we need a yes/no result, we don't need a yes/no result, we need a cutoff, represents potentially a perfectly valid hypothesis. I'm not a virologist. It makes sense to me that makes sense. It's a perfectly valid hypothesis. But at the moment, the technology we have for the analytical measurements may not be able to do it. And we need to remember this when we are just advocating these things. There is work in the standardization community going on, though, throughout the globe, really, working to try and improve standardization of before SARS-CoV-2 testing, molecular measurement, but now a lot of us are working on SARS-CoV-2 testing. There are external quality assurance methods. This is just 3. I'm sure many of you are familiar with them, which will improve the harmonization of those measurements. And we're also working on methods like digital PCR with very clearly defined standard operation procedures to try and provide reference measurement procedures that may support the value assignment of both reference materials, quality control materials, but also clinical samples. So we can look at those reference ranges that may, in the short term, certainly support the use of molecular methods like real-time Qpcr and the isothermal method, and perhaps in the longer term, actually work as diagnostic methods in their own right, because they are much more reproducible. And also we have the likes of NIBSC and the WHO working frantically to try and produce an international standard like the ones they produce for HIV and hepatitis C to support global harmonization of this measurement, to support both things like the limited detection measures and also potentially quantitative cutoffs. But we need to remember that the left-hand side of this slide is where all the chaos is. It's where the problems are. In the UK, we have sampling using swabs, nasopharyngeal swabs. They vary from an expert clinical worker to someone at home sampling their child. This is actually one that I had to sample my daughter. I'm not an expert in this. This is not a fun process. I would be very surprised if the sample I took was as good a sample as someone who's trained to do this. This is going to add variation. So in the last 4 minutes or so, I've touched here really on things that impact on the sensitivity, things that might give you false negatives. But there's also an increasing discussion around the false positives that many of you may be familiar with. And I just want to touch very, very briefly towards the end of the presentation on some ideas we've had as a group as to how we can front that. Now, one of the issues is that many of you may be aware that there are laboratory developed tests, or there are primers you can find or recommend, or not recommended, but referenced by the WHO. And you can get these to a range of different regions of the virus genome. And people have obviously done this across the world. And a number of them are publishing work that stating that these samples or these assays cross-react with non-SARS-CoV-2 nucleic acid. And this is leading to inconclusive false positives. I'm not going to mention these specific papers because I don't want to raise them, but these are what's being written inside, and these are peer-reviewed in cases. Other papers state that the observed unspecific signal may be due to dimerization of primers and probes or unspecific primer binding, polymerase activity. They rule out contamination because of sporadic measurements, because they were careful in setting their experiments up. And in this example, people are actually doing limit of blank measurements, as you might in an ELISA. They're having this background signal for the Rtqpcr, and they're doing 3 standard deviations above this to call this background. I've had conversations with people trying to implement tests, trying to understand how they can reduce their signal to background noise. When measuring SARS-CoV-2 RNA, there should be no background noise. The virus genome, as we've heard already today, some fantastic talks, is quite unique, certainly when compared to other commonly measured pathogens that you might be picking up in the respiratory sample. And the trouble is that, as well, these papers are alluding to, 3 of the 7 or so assays having this issue. Now, it's possible to design an assay that will cross-react, but it's highly unlikely in this context because so many people have looked at them, so many people have used them, and they haven't seen this issue. And the reality is, if you are designing an assay to something like SARS-CoV-2 and it is cross-reacting, then it needs to be redesigned so it doesn't cross-react. It doesn't need to be doing so. I think this is contamination. I think this is due to a number of factors. The fact that many people are trying to do assays, many people might be trying to apply these methods having never necessarily worked with PCR, or they have worked with PCR, but they've been doing genetic tests. So they're not used to very, very high cycle numbers, 40 cycles or more, and trying to call positive or negative results, nothing, as a negative sample. It's challenging. It's not an easy thing to do. And here's an example from Great Ormond Street, where there are 60 negative patients measured here. And there's a positive control, as you can see. And that is using the N-gene assay Catherine Harris runs, and an RNase P human messenger RNA construct control to just as a positive. Great Ormond Street also included a positive virus that they spiked, bovine distemper virus. And when they included the assay for this, they suddenly get half the signal coming up positive. And the reason for this is because the primers for the PDVH have the SARS-CoV-2 DNA in them. And the reason they have the SARS-CoV-2 DNA in them is because almost every oligo house on Earth is making these molecules. And we published we published an opinion piece recently on this very subject. And the fact that when you normally do a PCR, if you're a category 3 organism, you'll make the PCR product. But you should never make the PCR product in the same place you order your primers. That may no longer be a precaution we can take because everybody is making this molecule. So to conclude, looking specifically at the analytical performance of molecular detection, the technical error associated with SARS-CoV-2 diagnostic pipeline is a major source of variation and has a potential to impact on analytical sensitivity, the limit of detection. And the Rtqpcr CQ value, or CT or CP, whatever you want to call it, is currently too biased for a single cutoff to be broadly used nationally or internationally. We cannot say cutoff at 30. And this is not for clinical reasons, it's for purely analytical reasons. False positive signals are most likely to be due to variable technical artifacts, contamination being a prominent one, baseline drift. These are all issues that are not should not be accepted in a specificity measure as a false positive. We should be working around them. Primary cross-reactivity is really unlikely when measuring SARS-CoV-2 and arguably should not be accepted. There should be no background signal when measuring SARS-CoV-2 diagnosis using Rtqpcr. But the measurement standardization science can provide diagnostic confidence through the birth of AQAs and the work we're doing, and solutions to some of the challenges faced in the development and application of these diagnostic tests. And just a final point, that national measurement institutes exist globally to assist with these very challenges, but we are generally new to the biospace and certainly new to things like molecular biology. So we may not be so familiar, but the chances are there will be a national measurement laboratory that you have access to, or one you can make contact to if you don't have one within your country, who will be able to support and help in these circumstances. So I would just like to end by pointing out that perhaps some of the statements that we are hearing about a quarter of tests being false negatives or a third, we need to be practical about what we're expecting from these methods. And I would, having looked at some of the stuff we've been working with, I would not be surprised if the sensitivity of many of these methods is around the 60 to 70%, if not worse, depending on those pre-analytical stages. So we need to be a bit practical in what we can expect. I'd like to thank many people. I don't have time to go through all them all here. The work we do is global, and so we have some great collaborators internationally, and so I'm grateful to them. And I'd like to thank you for your time and attention today, and thanks again for the invitation to speak.
Thank you very much, Jim. So we received a few questions, but we don't have time for all of them. I'll try maybe to compile several aspects in one question, and that is to say that, I take your points about the different aspects of measurement and sensitivity and specificity. But as they say, perfect is the enemy of good. And if we were trying to optimize everything and do everything cautiously, we may not have been in a position where we can do millions of tests per day. And perhaps to curb a pandemic, we don't need to be very accurate, we just to be very active operationally. And this goes back to questions like cutoff and what is the public health action that is needed for various measurements. So what is your take on that? I mean, you come from the measurement side of things, but still.
No, I think this is a valid point, and I'm grateful for the question. Absolutely, we see this as well when people speak up and go, well, this is the perfect scenario, this is how things can work. But I would parry that, though, with arguing that we need to have an idea of the reproducibility. We need to have an idea of how our methods are working, and not assuming they're all working comparably, because as you've presented in many of your presentations, Jacob, we have the countries reporting different data sets, or, considering different groups of patients positive. If you include in that the analytical stages of the methods being highly variable, this is also going to add chaos. And so I think it's the work, this type of work, and the need for standardization to provide this confidence is important. But no, I'm certainly not advocating that we all have to have the most sensitive method possible. I guess I'm advocating that we should perhaps try to certainly consider issues around the reproducibility and how are we going to get those upstream stages working. And when we talk about cut, and as people have done in the UK on the radio, and I've thrown things at the radio, it's simple. We just have a cutoff. I think we need to be a little bit realistic in what we can expect from these methods, and think about how they can be used in that way. But no, I'm certainly not advocating we have to have the most accurate, depending on what we mean by that, analytically accurate, should we say, method. So no, that is a fair point.
Okay, thank you very much. And I will now hand over to Ed.
Okay, thanks, Jacob. So the second speaker today is Dana Wu. Dana Wu is from the Hebrew University in Israel. She is an infectious disease doctor, but she's also the head of the clinical virology. So she's taking care for patients with COVID-19, and she's also very much interested in trying to improve the efficacy of the tests. And the title of the presentation will be Sample Pooling. Dana, the floor is yours.
Thank you. Thank you, and I would like to thank the organizers for inviting me to give this talk. And this is, in fact, a great opportunity for me to share with you our accumulated large-scale experience utilizing sample pooling for the expanded detection of SARS-CoV-2. Sorry, why is it not working? Okay, so at the time of the emergence of the pandemic, our clinical virology lab has been a mid-sized diagnostic lab serving hospitalized patients and outpatients in the Jerusalem area and beyond. We were comfortably performing around 120 tests per year, combining viral serology, culture, and molecular assays.
Sorry, Dana, I can't see your presentation. Am I the only one, or?
Okay, sorry.
Share the screen, please, again.
Sorry. Share screen.
Great, now we see the
You
slide.
see it now?
Yeah.
Okay, have you seen this slide already?
No slides were shown.
Okay, so I'll start from the beginning. So at the time of the emergence of the pandemic, as I've told you, we were a mid-sized clinical virology lab, comfortably performing around 120K tests per year, combining viral serology, culture, and molecular assays. We were steadily expanding our Rtpcr assays and the use of robotic systems, which in fact proved to be quite helpful upon the emergence of the pandemic. Our research is focused on ex vivo modeling of viral, mainly HCMV infection in human target tissues representing the viral entry and transmission sites, and on new antiviral drugs. And in fact, we have recently expanded these studies to include SARS-CoV-2, but I will not talk about them today. So I guess like many of you, we have been catapulted by this pandemic into the high throughput arena, turning very quickly into a SARS-CoV-2 testing mega lab. And here you can see our weekly distribution of tests. It's quite fluctuating, overall performing around 300K tests during the last few months. And our tests come from diverse sources. Our samples come from symptomatic outpatients, hospitalized patients, and importantly also from screened healthcare personnel, nursing homes, and essential industry workers. And looking at the SARS-CoV-2 diagnostic workflow, already well described by Jim, this process is pretty familiar to us from the diagnosis of other pathogens, but there were some peculiar features and challenges here. Apart from the specimen safety issue, which proved to be quite a considerable bottleneck, some main challenges including the need for high throughput, short turnaround time, with rapid exhaustion of reagents and equipment during the pandemic, and also importantly, the increasingly recognized need to detect not only symptomatic, but also asymptomatic infection viral carriers to contain viral spread and guide control measures. And lastly, we had to deal with the fluctuating diagnostic activity over time, largely impacted by the changing prevalence of infection and by the changing testing policies. And this brings me to sample pooling or group testing. And I will discuss some general aspects, but mainly tell you about our practical experience utilizing sample pooling to increase our testing throughput, to address these peculiar challenges, and particularly to expand screening of asymptomatic individuals. So by now, I guess many of you are familiar with the main methods of sample pooling, so I'll be very brief, just mention the most relevant methodologies here. So the first and the most simple and straightforward and most frequently used is the Endorsement Pulling. Here, an equal number of samples is mixed and tested in pools in the first round. All samples in negative pools are declared negative, and all samples in positive pools are retested individually in the second round. So this is a simple scheme, but there are more advanced versions of Endorsement Pulling today with potential additions of extra rounds entailing some extra time, or dimensions entailing some extra complexity to increase efficiency and group sizes. Now, these are the more complex, innovative, but less frequently used methods, so I'll not elaborate any further on them. So coming back to the simple scheme, one has to keep in mind that the potential drawback of the two-round method is some delay in confirmation of positive results. So an alternative pooling method involves the performance of all tests in one round, distributing the samples into a matrix of overlapping groups. As you can see, each sample is concurrently tested in several pools with varying degrees of complexity and efficiency. So again, there are different degrees of complexity here, and an example of a simple version. I'll just show you briefly our preliminary experience when we pulled 75 samples into 3 5-on-5 matrices. It was a successful experience, but we did not further continue in this direction. Again, there are more advanced and complex versions today employing combinatorial pooling strategies with special software, and some of them are right now in pilot clinical trials for the detection of SARS-CoV-2. So moving to sample pooling in the context of SARS-CoV-2, there has been some initial hesitation or concern over potential expected loss of sensitivity when you dilute samples, when you pool samples. Recent validation and proof of concept and early implementation studies were conducted by us and by others. And the majority of these studies employ Endorsement pooling of 4 to 32 samples per pool. And all these reported studies generally involve hundreds to a few thousands of tested samples, low positivity rate over a limited time period. One should bear in mind again that the different protocol and stages of pooling used made quite difficult to compare between these studies. However, one common feature is that despite some expected loss of sensitivity, all these studies have shown sufficient diagnostic accuracy. And perhaps reflecting the increased confidence in pooling, the FDA has recently issued the first EUA for sample pooling in diagnostic testing recently in July. So with this, I would like to move to describe our implementation of pooled RNA extraction and Rtpcr for SARS-CoV-2 detection. So we pull samples in this stage of the process and share with you some important lessons derived from a large-scale pooling experience, spanning more than 124K samples tested by pooling. So early during the pandemic, we assessed several pooling strategies and eventually combining considerations of predicted efficiency, accuracy, and ease and simplicity of operation, we chose a Dorfman pooling strategy of 8 samples per pool. So why 8? Here you can see the predicted efficiency of Dorfman's pooling. It's a calculated theoretical measure showing basically how many more patients or samples can be tested with n sample pools compared to individual testing. And then based on this calculation, we predicted that an n of 8 sample pool would assure a reasonable efficacy over a range of prevalence rates. We also predicted that this modest pooling size would not significantly compromise our sensitivity. And indeed, in early studies, validation studies of 8 sample pooling, and this has been published already, we showed that 8 sample pooling maintains clinical sensitivity, allowing detection of single samples with CT 34 to 36. Jim just described the CT concept, and missing only very low viral load samples of probably negligible infectivity. We also showed that under very low prevalence conditions, there was a significant increase in our throughput and efficiency. And as for the practicality considerations, which I mentioned, we use an easy robotic setup. In fact, these pictures are taken from a short movie that you can watch under this link. And looking at the process outline and timeline, eventually what we can test thousands of samples by pooling in less than 24 and most frequently even less than 12 hours. One thing that I would like to emphasize is that you cannot plan on a large-scale pooling without a special software. And indeed, together with our IT team, we have developed a validated software for the convolution of the data and automated reports. So moving now to the large picture, and I would like to tell you that the ongoing sample pooling over time presented us with some challenges of fluctuating prevalence rate. So we had to adapt, we had to be dynamic in our approach. Now this is a complicated slide, but I will simplify it. So in blue, you can see the distribution of numbers of samples tested in 8 sample pool. The black line demonstrates the positive sample rate. And what you can see is that the positive sample rate really steeply increased in July, for example, and we had to employ adaptation to maintain our efficiency. So we switched to 5 sample pooling, here shown in the light blue. And thereof, and since then, we had to adapt this alternating, adaptive, dynamic pooling approach to maintain our efficacy. So overall, this is the distribution of our pools. You can see the pools of 8 samples, pools of 5 samples. The majority of the pools remained negative despite the changing prevalence. What you can also see is that drilling into the positive pools, we saw that while the majority of them contained only 1 positive sample, a significant proportion, quarter to third of the pools contained, of the positive pools, contained 2 or more positive samples. And why am I telling you this? Because this may have implication not only towards the efficiency of pooling, but also towards the sensitivity of pooling. Let me explain. The large perspective allowed us to take a closer look at the actual loss of sensitivity CT increase of 8 sample pool. So comparison of the pool viral CT in the y-axis with the individual sample CT for quite a large number, 800 single positive samples identified within pools, demonstrated approximately a 3-cycle increase. You can see it by regression analysis. Now, nicely, this actual observation in practical large scale completely corresponded, it was in complete agreement with the predicted CT increase. But this completely changed when we had, in the presence of 2 or more positive samples in the pool, suggesting a potential interplay between the prevalence rate and the sensitivity of detection by sample pooling. Let me explain. The presence of 2 or more positive samples, here shown in the light dots, as you can see, shifted down the pool CT relative to the individual sample CT, increasing the sensitivity. The pool CT value was mostly defined by the high viral load sample within the pool. Under the circumstances of multiple positives within pool, as you can see here, for example, low viral load samples benefiting from the strong partners in the pool can thus be detected with greater sensitivity. As the infection prevalence increases, there are often 2 or more positive samples in the pool. Thus, increased prevalence may increase the sensitivity of our pooling detection. And finally, this is my last result slide, calculating our actual empirical efficacy measured by the total number of samples divided by the total number of PCR reactions. We found that our empirical efficiency over a large scale by practice is in fact better than the predicted efficiency. So the efficiency for pools of 8 was 4.6, better than the predicted 4.1 for the observed prevalence, and the efficiency of 5 sample pools was 2.3, again, surprisingly better than the predicted one. So altogether, in all pools, we performed 30K reactions to test 124 samples with efficiency of 4.2, saving 75% of our reaction, 94K reactions to achieve the same number of results. So in summary, we have employed and monitored large-scale 8 and 5 alternating sample pooling over time. Large-scale data over 120 pooled samples revealed a high empirical efficiency of sample pooling demonstrated in practice for quite a wide range of prevalence rates and overweighing the minor clinically insignificant sensitivity loss. Ongoing dynamic and adaptive sample pooling approaches, as I show you, can maximize resource saving under fluctuating infection prevalence, as long as the prevalence is not beyond 5% and more. And then our empirical evidence strongly projects on the wide feasibility and benefits of carefully conducted sample pooling in the current pandemic setting to enhance continued surveillance, control, and community reopening. And with this, I would like to acknowledge my partners. Actually, I don't know where to start, so I would like to just acknowledge all Hadassah Hebrew University Hospital Corona Lab members. I will just mention individually Dr. Esther Oikninjian, the supervisor of the lab, who orchestrates this activity daily, nightly, hourly. Dr. Moran Yasur, who has analyzed and summarized much of the large-scale data which I have showed you together with his student. Amir Peri, our IT manager, who made it all possible with dynamic softwares, and Professor Yuval Dor and team, who were really playing a major role facilitating this operation. And I thank you for your attention.
Okay, thank you, Dana. We have time for one short question, I think. And that's the question on the environment. So do you think that this pool testing can be done in each laboratory in just a normal laboratory environment, or do you have to take special precautions to prevent contamination?
Actually, I think, well, it requires adaptive capabilities, good validation capabilities. The endorsement pooling is quite simple to perform. You need a good software, a good robotic system, and we haven't seen, in fact, contamination using the pooling. I think it's quite a high proof, because once you get a positive pool, let's say it's a false positive pool, we actually make a very strong point of being very sensitive with the pooling. We haven't seen too many false positive pools, maybe 100 in all the tens of thousands that we have done. But then when you deconvolute the pool, open it, you retest all the samples. So it's a double check against contamination.
Okay. Okay, thanks again, Dana, for your excellent presentation. Then we go to the last speaker of today, and that is Corine. Corine is a medical microbiologist, virologist at the Erasmus University in Rotterdam. She's very much interested in immunity against viral infection and did a PhD thesis on immunity against influenza virus. And she's now very much interested in the humoral immune response to SARS-CoV-19. Okay, Corine. Corine, you are muted. Corine, you're still muted. Can you unmute yourself?
Sorry. Like, is it okay? Oh, sorry. There we go. Okay, there we go. Hope it's okay now. Yes. So thank you, and I would like to thank the organization for the opportunity to give this presentation on immunity against SARS coronavirus. First of all, I think it's very important that although in a diagnostic setting we mostly measure antibodies as a parameter of protection and as a sign of immunity against COVID. It's very important to realize that, of course, the immune response consists of multiple pillars, and that, for example, T-cell immunity, but also other pillars, well, are important to keep in mind. But nevertheless, in a diagnostic setting, antibodies are main and important to get an idea about the immune response. So when we look into the antibody isotypes, which we detect against SARS coronavirus type 2, we see that the IgM and IgA antibodies actually come up quite rapidly within the first 10 days post onset of disease, as we can see in the top graph. Whereas IgG antibodies have a much much larger variation. Sometimes they come up directly with the IgM, but we also see patients in which antibodies come up after 30 or 40 days. So that's quite a variable parameter once we are measuring in serum. Once looking into the height of the titers of IgM, IgA, and IgG, we see that both IgM and IgG make a significant increase very early after onset of disease, of course, as expected, whereas IgA will quite rapidly disappear. And IgG remains to increase quite steep after the first and second week. So once we're talking about the height of the titers. What is quite interesting in COVID-19 patients is that there's quite a big difference in the amount of, so the robustness of the antibodies depending on the severity of the patient. So what I show here are the first two graphs which we obtained from COVID-19 patients in the beginning of the pandemic. In red, we see the severe patients, and in green and black, the mild patients. And the interesting thing is actually that these trends are still being seen in the large studies which have been conducted nowadays. So we really see that the severe patients have a fast and robust response, whereas the mild patients need more time to develop the antibodies, and generally the antibodies titers will remain lower in the mild patients. When measuring these antibodies in a diagnostic setting, most of the commercial assays which are being used target either the N or the S antigen of the virus. The N antigen is obtained from the nucleocapsid protein, which surrounds the viral RNA, whereas the S protein is the spike protein, and it's the characteristic spike of the virus. And this spike protein is also the basis of a lot of the vaccines which are currently being tested in different trials. When we look a bit further into the spike protein, we see that there is an S1 and an S2 domain, and this is what we also find back in the diagnostic tests which are being provided. And interestingly, the receptor binding domain. This receptor binding domain is an interesting part of the epitope because it correlates very well to neutralization. And that's what I will show further on. So before going into these tests, I think it's extremely important to very well define the aim of the serological testing prior to selecting an assay or choosing the assay which you want to use. For example, in our hospital, we look into the infectivity of individuals, and we want to see if an individual is protected. But of course, when looking in the population, you might think about herd immunity, and you just want to know if somebody is exposed. On the other hand, if you want to use plasma products of people who recovered from coronavirus, you want to really know how high the functional antibody titer is. And then, of course, you have people interested in home testing, where it should be accessible and easy, but still reliable. And nowadays, we are interested in testing vaccination responses. But all these different aims may need different characteristics and performance characteristics. So we started in the beginning of the pandemic in being interested in infectivity of individuals and how does their immune response work. For that part, we thought it was very important to look into virus neutralizing antibodies, as these are the functional antibodies of your humoral system. And for that, we use the plaque reduction assay, which I think a lot of people know. It's an assay in which you dilute the serum, you add the virus, the combination of serum and virus is added to a cell layer. And by this way, after 8 hours of incubation in our setting, we can look how much infected cells there are, which is reflected by the amount of plaques of the cell layer. You then get a titer, which corresponds to the amount of plaques. So this is an assay which we have used a lot to look into the immunity. And like I already showed in the first slide, when you look into neutralizing antibodies as well, we see that the hospitalized severe patients very rapidly get very high titers of neutralizing antibodies, whereas the mild patients take more time, but still develop quite adequate levels. Most of them, of course, there's always people who are an exception on this, this aspect. The next question then is, if we look into functional antibodies, what does it say about infectivity of the virus and then of the patient, let's say? For that, in the first months of the pandemic, we have cultured all the respiratory specimen which we obtained from the hospitalized patients. After 2 days of putting the specimen on a susceptible cell line, we checked with a fluorescent staining if there was indeed infection of these cells. And what we saw is that it's quite expected, but we saw that there was we could only culture these viruses once the RNA copies were high enough. And when we then look into the talk of Jim, who just said it is quite hard to get this strict cutoff. Well, we found that above a cutoff CT value of 30, we could not culture the virus anymore. And of course, you have problems with the transport, and there's a lot of variables. But for all these patients, we tried to put the sample on the cells as quick as possible. After we obtained the sample, they were picked up at the ward and directly put on cells. So yes, we saw that, okay, above this certain threshold, we cannot culture the virus anymore. But just putting a threshold for this PCR did not seem enough. So what we wanted to know is if we could add a parameter like the serology. And what we saw is that in all these patients in whom we could not culture the virus anymore, the amount of antibodies were above a certain threshold as well. So when putting this in a probit analysis, we saw that above a titer of 1 to 40 in our neutralization assay, there was no patient in whom we could culture the virus. Of course, again, when using this kind of neutralization assay, you would need some standardization, and currently we are working with standardized sera to test and to see if we can quantify this better. But it's just an example of how we have added in our center, we have added these serology parameters to the diagnostic workup to define if we think that a patient is still effective, yes or no, despite the PCR, which remains positive for a very long time. We think it's very important because the the effect of isolating a patient is quite big, and we do not want to do that if we do not think that the patient is effective, if it's not effective anymore. Of course, a virus neutralization assay is very labor intensive. You need a BSL-3 lab. So what you would want is a good diagnostic assay which correlates well to the virus neutralization. For this purpose, we have compared 2 manual ELISAs and 3 automated serology systems to our virus neutralization data within the different severities of disease. And we actually found that it was quite hard to find a good correlate. In our hands, the one-tie IG total ELISA was the only ELISA in which we could set a cutoff. So we increased the ratio cutoff to 10, because above this ratio, we saw that all patients indeed had neutralizing antibodies, but it's still not perfectly linear, and it's something we are working on to improve. It should be realized that once we are doing this kind of analysis, that of course the one-tie ELISA, it measures a total immunoglobulins, whereas other assays might measure only IgG. And it's important to realize that in a virus neutralization assay, we measure all neutralizing capacity, so also the short-lived IgM antibodies. The question is, again, is that what you want to measure? Are you interested in these short-lived antibodies, or do you really want to measure only the long-lived IgG? Something to take into account, I think. That brings me to the next question. Now we know how to determine the antibodies. It's very important to think about what does it mean, and how long does it protect people once you detect these antibodies. And of course, this longevity of the response is something what you need in actually all questions you want to answer, specifically when looking into vaccination response or protection of the individual. Well, we have several papers which have come out, and this was one of the first papers, and it received quite some attention in the Dutch media because it shows that within 100 days there's a severe decrease of antibodies, both IgM, IgA, and IgG. And here they show both the RBD and the N tests. A more recent paper in which they tested the population of Iceland does not show this decrease, and it shows that actually the responses stay quite stable in recovered patients from COVID-19. I think this illustrates very nice that the studies looking into these topics need to be very well defined, and in order to compare them, well, the assays and the severity of disease and the population and the background has to be very comparable in order to really make your conclusions. I think a quite nice summary was made in an editorial in the New England Journal recently. I think it very nicely summarizes how we think that it works in SARS corona immunity. Because in blue, we see those short-lived, fast response with antibodies within the first weeks. This is mainly caused by the short-lived plasma cells, but this is expected to wane, so it will decrease. And at that time point, you will get the long-lived plasma cells who take over, and only few of these long-lived plasma cells will be enough to really make a reduced functional response once there's again exposure to the virus. So I'm not sure if the problem is really in this decrease of antibodies, which can be observed and which is likely to be observed. But the problem is how will you be protected once you re-encounter the virus? And is that good enough to protect you? Probably it's not good enough to induce a sterile immune response, because that's what we see with the other circulating seasonal coronaviruses. You will expect that people get reinfections, and probably hopefully this will be mild reinfections, and the immunity which you obtain, hopefully will be enough to protect you from a severe pneumonia or another lower respiratory tract infection. I think an important question there is, how to prime the people. And if that could be done either by a vaccination or multiple vaccinations, or a real life exposure like people are experiencing now. Yeah, I think that's something which is interesting to follow up in the coming months, and specifically in the vulnerable populations. So is this recall immunity good enough for protection of the vulnerable population? So we've seen the first documented reinfections. Up to now, we indeed see that these seem to be mild infections. So unless that these are severely immune compromised patients in whom the humoral immune response is already not yet good enough or not present at all. But I think we can expect more reinfections, and it's important to follow up on these reinfections and yeah, see what the immune response does in these patients, not only the humoral immune response, but of course as well the T-cell immunity, as it might be a very important component in vaccination as well. So that's what I wanted to talk about. So in conclusion, the antibody levels against SARS coronavirus 2 largely depend on disease severity. The aim of serological testing is very important once you select an assay, and I think it should really be part of the diagnostic choices. We think that adding serology to a diagnostic workup of patients who are admitted to the hospital can really support infection prevention measures, although additional commercial assays will be very helpful in that regard. Finally, well, it will be a very interesting period to see how the ongoing studies on SARS corona immunity will define the benefit of, for example, herd immunity and vaccination responses. With that, I would like to thank you very much for your attention, and I would like to thank all my colleagues at Erasmus within the Netherlands and outside for their collaboration. Thank you.
Okay, thank you very much, Corine. There have been several questions in the chat. I have selected 2 questions. The first question is on what do you know about the antibody response in patients who are treated with steroids and with antivirals?
That's a very interesting question. The responses in antivirals is something we are following up on currently. I do not have very much data on that available yet, because the large-scale treatment, well, and the long-term follow-up is of course something which is currently ongoing. But for these patients, we do not expect it to be very much affected, because they will have had their initial priming. Okay,
and then a very important question, Corine. So you clearly demonstrated that there was an excellent correlation between your neutralizing antibodies and the antibodies measured by the one-tie assay, and that are the antibodies directed to the receptor binding domain. So is that the favorite antibody that we should test for vaccine efficacy?
I think that will be a very important one, yes, indeed, because most of the vaccines are using the S domain as well. So yeah, I think that will be a very important part, besides the T-cell testing and looking into the T-cell responses, because I think that will be a very important component to take along in when evaluating vaccination responses. But again, less easy to do on a large scale.
But I mean, if you have to select one of the 5 assays that you demonstrated, so the one-tie, the liaison, the other assays, only the one-tie assay measured antibodies against RBD. All the other assays had the spike proteins or the nuclear proteins.
Yeah.
So do you still recommend that, despite that the vaccine is containing the S protein, the S antigen, that you measure the antibodies against the RBD as a matter of vaccine efficacy?
Yeah, I would definitely take it along, but of course, for measuring the vaccine efficacy, we will do multiplex antigen testing first. So yeah.
Okay.
It should be part of it, definitely, yeah.
Okay, thank you very much. Yes, thank you very much, Corine. On behalf of Jacob Morin, I want to thank all the invited speakers for the excellent presentation and especially also for keeping on time. And I would like to thank the audience for listening to us and asking all those interesting questions. Thank you very much.